Okay, so what we'll do is we'll share the screen and we'll start the process of learning that we essentially call reverse vaccinology. Okay, so the ultimate goal is to understand about, okay, vaccine design, delivery and all such things. So when we talk about vaccinology, what we are essentially speaking about? We are essentially speaking about things like the main three things that we are about to talk. One is vaccine design, how to design a vaccine, okay, then optimizing the shelf life, optimizing the shelf life of the vaccine, okay, and the other thing that we are interested in is to enhance potency. Okay, so these are the things we essentially talk about when we are talking about vaccinology. I am audible, right? So there are some interesting things as well. For example, first of all, we should understand what exactly are vaccines and how do they work. So vaccines are nothing but there are multiple types of vaccines that I think you are already aware of. Okay, some types are some of the types are say subunit vaccines. Subunit vaccines. Okay, these are essentially genetically engineered vaccines. Okay, then there are live attenuated vaccines, and there are killed vaccines. Okay, or killed vaccines. So what is the primary objective of a vaccine? The primary objective of a vaccines are nothing but what are the primary objectives? So the primary objective of a vaccines are to enhance the what do you call to enhance what we call as adaptive immune response. Okay, adaptive immune response, okay, which is acquired. It's an acquired what do you call trait. Okay, it is not by default. So vaccinology or vaccines, the main goal of vaccines are to enhance the what do you call is this is the goal of vaccine. Okay, just hold on. So this thing of vaccine is to enhance the adaptive immune response. Often by and large this is very interesting, this is mainly given to vertebrate animals. Okay, this is very interesting. Vertebrate animals, you cannot give a vaccine to invertebrate animals. Okay, of vertebrate animals. Okay, this is important. Okay, so the other things are why we are talking about the design, because the design will determine the type and delivery mechanism of the vaccine. So vaccine design is important in order to determine what will be the type and what will be the delivery mechanism of the vaccine. Okay, for example, what we are trying to say when we talk about delivery mechanism, whether it is whether it is delivered with an adjuvant, whether delivered with adjuvants or just like that, okay, or the route of delivery, for example, whether it will be injected, it will be oral, okay, the mode or form of the vaccine, dosage form, such as whether it will be delivered orally or IM or IV. So those considerations are being determined by means of the design of the vaccine. Okay. So this is important. Then there is one more thing, optimization of the shelf life. So this is again very important point. Okay. So here the important point is, okay, what is the important point? That how it will be preserved. Okay. Whether it will be preserved at 4 degrees or less than that. And one more thing, the expiry date will be how much, that also is determined by what they call this optimization of shelf life. Now enhancing a potency, this is very important. Okay, enhancing potency means whether if the vaccine is so the king has admitted. So what is this thing, that if the vaccine is of single target or multi-target. Okay. Let's give an example. Okay. So what could be a good example? Example could be like polio. Okay. Single target vaccine, whereas if we call as what, DTP, okay, it's a multi-target vaccine. So these are important considerations that you should have. Okay, so we'll reduce the size so that it is accommodated here. So these are very important, what do you call, considerations. Okay, so these are very important considerations. Okay, so today we will discuss multiple different things. Let us give you this thing of this particular research project. Okay, so I guess I'm audible. So here what happened, we were given a task to identify some uncommon epitopes on the uncommon epitopes in order to make a vaccine which will strongly bind with MHC1. Okay, so this should be discussed first. So as you can see here, this is a international group. This fellow is from, I think, Nigeria, and this is from Gujarat, this is from New Delhi, this is from Ukraine where this thing is going on, this is from Uganda, this is from Cairo, Egypt, this is from Manila, Philippines, this is again from Nigeria, this is also Nigeria, this is also Nigeria, and this is me. Okay, so it's a kind of international group. So what we were looking at, that this study was being done when the COVID vaccine was being developed. Okay, it was full-fledged going on, and I think the first trial was also going on. At that point of time, we started this work. So we found out one thing, that there are some patients who are not responding to the vaccine. Despite of giving the vaccine, people are not becoming much stable. So the reason behind that was the basic understanding of the COVID virus biology, that it's a RNA virus and therefore it will be having random mutations, and eventually it will be not very much effective. And after we understood this, we also tried to figure out that is there any more epitope which can be explored for vaccine, which will be more stable. So that was the idea. So this is the abstract only. This is a large paper, and as you can see, it was been supported by Chan Zuckerberg Initiative. Can anybody give us some idea what is Chan Zuckerberg Initiative? Anybody has heard of it among you people? Who can tell me what is Chan Zuckerberg Initiative? Anybody has any idea? If you can unmute. I am audible, right? You have any idea what is Chan Zuckerberg Initiative? If you are sticking to this domain, a biomedical domain, so you should understand that who are the potential fellows who can fund your research or your study or whatever. Okay, so who can tell me what is Chan Zuckerberg Initiative? Any one of you? So anyway, as you can I think figure out that he is the son of Mark Zuckerberg, and upon his name his father has initiated one what shall I say, kind of like what shall I say, a fund which has been given to throughout the world for various purposes, by and large for research. Okay, and this is again Cold Spring Harbor Laboratory. That also you can Google and you will find out what is Cold Spring Harbor Laboratory. Now this is the laboratory from where n number of Nobel Prizes, including the Nobel Prize for PCR. Professor Kary Mullis, he is also belong to this Cold Spring Harbor Laboratory. So you could have these ideas. So this is the full text. We'll just see what exactly is there, so that it could give you a good idea. So this is the introduction where we have spoken about how this COVID-19 has originated. Then we are talking about the protein sequence. I will show you some figure. If okay, now these are some images and tables. This table, okay, this is the whole this thing analysis. Okay, next to that, if there are some images, it will be good for you to understand. Okay, here you see. So here what happens, these are the proteins which has been identified on COVID-19 virus surface glycoprotein, which are not very exposed. Okay, these are internally made. That means what happens now, it's a three-dimensional structure of the protein, and some internal part of the viruses are having these proteins. So they are not exposed, and eventually they are not very prone to mutations also. Okay, you can enhance the figure as well so that you can understand nicely. So these are the secondary structure, these are the tertiary structure, secondary structure, tertiary structure, and you can see these points which are highly stable regions. Okay, despite of COVID is a RNA virus, these regions are somewhat, what do you call, stable. Now what is this? In this figure it was been identified that which is the HLA which binds with this particular domain very strongly. So we'll learn about that, what is HLA. So here what happens, let us consider this only, since the structures are there. Consider these proteins. Okay, so I'll tell you from this protein what exactly is HLA. Let us take this protein. Okay, so we'll take this protein. Okay, consider that this is a protein. Okay, Now this is where it is present. It is present in the COVID virus itself. So consider that this is the COVID virus and this is the corona. Okay, the surface glycoproteins. Okay, this is a hypothetical drawing. Okay, do not get very serious out of it. Okay, so consider that this particular protein is a hidden protein and it is present somewhere here. Okay, where it is comparatively what you call hidden. Okay, why hidden? Because in the rest of the area there are a lot of water molecule, everywhere water. Okay, it's a watery environment, aqueous environment. Okay, so these are all water molecule, let us say. Okay, H2O. Okay, O. Okay, let us say these are all water molecules. So it's a aquatic environment or aqueous, because everywhere it is water, right? Any living being somewhere present means it is in water. Oh, just hold on. So these are all water. These are all water. These are all water. Okay, but this particular protein, no, that particular protein domain, or let us say motif, this particular motif or the domain is remain hidden. Hidden domain. Hidden domain. Domain on the अभी क्या आ गया यार, hidden domain. Okay, where it is present somewhere here. So let us give him entry also, so that he feels himself, he considers himself very lucky. This kind of teaching is I think very rare, if not very frequent. So what happens, this is a hidden domain which is present on the COVID virus only. Okay, let us write it down, COVID-19 virus. So this is COVID-19 virus. Somewhere there is a hidden domain. Now we have identified this domain and now we are trying to figure out how MHC1 molecules are binding on it. Okay, so what is the rationale, that also we should understand. Just hold on, I will give you the rationale for you. Not this, we'll put this. Okay, so what happens, consider that this is a cytotoxic T lymphocyte, or shall I say how, TC cell. So what is TC cell? We have already spoken about it. TC cells are nothing but lymphocytes that mature in where? Thymus gland. Okay, they mature in thymus, and what is their role? Their main role is to help in cell mediated immune response. Okay, they are responsible for cell mediated immune response. Now what is cell mediated immune response? It's a type of adaptive immune response. Okay, so it is nothing but a type of adaptive immune response. It is not this thing secondary, it is primary. So adaptive immune response, it's a type of adaptive immune response which is taken care by means of cytotoxic T lymphocytes. Now what is their work? Cytotoxic T lymphocytes are essentially lymphocytes which are having nucleus. Okay, so it is having nucleus. So this is what happened. Okay, so it is having a nucleus. Okay, beautiful nucleus is there. So what happens, on the surface of this TC cell there is a receptor called MHC1 receptor. So what is the function of it? This MHC1 receptor is a receptor which will present viral antigens on the surface so that immune response will be started. Okay. So what it will do is it will bind this MHC1 on the surface of the T lymphocytes, will bind with a viral protein. This is a viral protein. If it would have been a bacterial protein, MHC2 would have been activated on some other type of cell. So we will write it viral antigen. Okay. So viral antigen, this is a viral antigen. It will bind with the MHC1 molecule on the present on the cytotoxic T lymphocyte, and this binding will eventually lead to what? Influx of all other what you call phagocytic cells. Okay. So it leads to what? It leads to the cell mediated immune response. Cell mediated immune response means the antigen will be killed. At the same time, the virus itself, where this antigen tag is there, it will also get killed. Okay. So this is the whole idea. At the same time, it will also produce memory T cells. It will also produce what? Memory T cells, for which memory T cells it will produce, and which will help on what? It will help on future viral attacks. Okay. It will help against, to tackle, let us write tackle future viral attack, okay, of this particular protein only. So where this particular protein will be there, it will be present in the COVID virus only, because we identified this area from COVID itself, isn't it? Okay. So memory T cells will be generated from the T lymphocytes only. So what will happen is, this T lymphocyte which binds with the viral protein will break down into two things. One is memory T cells, and another one is what do you call effector T cells. These effector T cells will kill immediately effector T cells. So what is the idea? The idea is to find out whether this protein is having sufficient tendency to bind with MHC1. So for that, what it should have, this viral antigen should get classified as an antigen first. So we shall first identify whether it's an antigen or not. Antigenicity. We shall find out what? We shall find out antigenicity. So in order to find out the antigenicity, what we should do, that is very important. Okay. So this is the whole thing. So what we'll do now, we'll do assessment of antigenicity first. Okay. So for that, yes. Oh no, not this. Linear antigenic epitope, absolutely. So what we'll do is, we'll find out the antigenic epitope, okay, by using this particular tool, and eventually we'll figure out whether this condition is met or not. Okay. So first and foremost, what we should understand that whether it is able to activate the T lymphocytes, and then whether it will be effective to produce the memory T lymphocytes. Okay. So a vaccine is a vaccine when it can elicit the response of, or when it will able to produce this. Okay. A vaccine is only a vaccine when this situation is there. That means this particular protein, which is a hidden domain, can only be a vaccine only because if it is able to elicit memory T lymphocyte production. Then only in future viral attacks, it will have the potency to tackle the whole infection episode. Okay. So this is the very basic understanding of how the vaccines work. Okay. Okay.